with scientists. I mean, with the sciences. I mean, we often think erroneously that science is one big thing. And so today we're able to unpack that a little bit around the work of genetics in the genetic era. So let me properly introduce our panel members. To my left here is Dr. Amy Lampazzi, who earned a PhD from Rice University in mathematics with a focus on the theory of knots. Dr. Lampazzi is also an alumna of UHCL's math graduate program and currently works at UHCL as director of the Mathematics Center and as a math instructor. She's always interested in biology. She minored in life sciences at the University of Missouri-Rolla, and as a graduate student participated in the program in mathematics and molecular biology. And to Jerry's right is Dr. Darrin Leleux, who is a veteran of the aerospace industry, who has been with NASA's Johnson Space Center for the last 16 years. He has worked as a space shuttle flight controller and now serves as a project manager at NASA. Dr. Leleux holds a PhD from Rice University in electrical engineering and is an alumnus of UHCL's Master of Science program in computer engineering. And he is also a teacher, an adjunct teacher in computer engineering department at UHCL. His current academic interest is in studying the intersection of the humanities and the sciences as expressed through art, philosophy, and literature. And I want to give a special thanks to Dr. Kevin Phillips, who has stepped in on behalf of our other participant who had a family emergency. So Dr. Kevin Phillips is an assistant professor in the Genomic Medicine and Diabetes Research programs at Methodist Hospital Research Institute in Houston and in the Department of Biochemistry and Biology at the University of Houston. His research group uses genetic tools to investigate how hormones such as thyroid hormone regulate metabolism and cholesterol homeostasis. So let's give a nice round of applause for So the first question I'm going to raise with our guest of honor, Dr. Geraldine Ondrizek, is what will this new genetic revolution bring to people, to life, to research as we know it? I mean, we live in a new era of genetics and biogenetic identity. And then she's going to introduce her work that very well answers this question in relation to the way that she's been looking at these big questions. And then I'd like each of our panel members to respond to this big question. I've made it broad enough, but specific enough so that you can address it in your own way. So from here, I'm going to leave it with Geraldine.
Can you hear me? Good evening. Thank you for having me. And,
can you all hear? Can you hear?
Yeah.
Good.
The first thing I'd like to say is that my work is greatly influenced by the smart people who are here at the table, the PhDs in science. Jane explained that she hosted an exhibition of mine quite some time ago in which I was documenting nature. I've been looking very closely at nature for a very long time. And at the time, it was looking at trees. And in some ways, my looking very closely at a cell or an egg, a spider egg, as you see in the film, is very much like the meticulousness that I would use in drawing. I'm now not just looking at trees as physical form, but now that I have equipment, the optical equipment available to me, be a scientist, I'm able to look closer. Work was looking and documenting all of the orchids of Southeast Asia and translating their names from the Greek and Latin to English to understand what I was looking at. That those labels of the flowers explained to me that they were not the labels, they were not saying where the plants were from or anything about the plants, but it was the scientist's name. I became very interested in the Linnaean classification system and Darwinian selection and understanding that we had a society that put a grid of collectors' chambers, one might call them, building furniture. I grew up with a father as a furniture maker and learned how to build at a very young age, but I got very interested in museology, cataloging, and categorization, and began, of course, to look through the microscope as well. One of my deep fascinations with the microscope is that the invention of the optical lens by Antony van Leeuwenhoek in Holland, and then the microscope by Robert Hooke, was originally in order to do thread count and look very closely so that they could understand the quality of the cloth that they were selling. But it was by chance that they actually saw bacteria swimming in water. So I find this really interesting, and I have also engaged, as you can see in my exhibition, with working with material semiotics. That is, I like to use a lot of cloth and physical form. So what you're seeing in the exhibition is my desire to turn science into part of life. The chromosome painting piece is printed onto silk. The spider film is within a handmade paper enclosure. So that's often how I'm trying to translate between art and science. So I'm not sure I can completely answer Jane's question, but the desire for one to make the scientific discoveries readable and palpable to people around them, I think, is to put them out visually in a way that they can also experience them, because this is a secret world in a way that scientists are privy to. This is one of the first drawings of cell division, of chromosome splitting, meiosis, and mitosis, and I hand embroidered it, discoveries documented on them. This drawing happens to have been done by a female scientist. Also, I'm so happy to have you here, Amy, because there aren't a lot of women in the laboratories, and there certainly weren't a lot of women at Carnegie Mellon in the sculpture studio either, so very interesting. The next thing that happened in my life, from a personal standpoint, is I had a child with a genetic anomaly, and that really made me have to do a lot of genetic research and also changed my whole body of work. And after the subsequent loss of that child, my husband and I went through a lot of research. We did have a very healthy child, but the knowledge that we gained after the fact really has made me compelled to continue to understand what's the cutting edge of genetic research and why do we need to know it, or the cutting edge of science in general, so all sciences. But I was invited to the Wellcome Trust as a artist in residence in London, England at Gasworks, and this is the DNA sequencer that did the big job of sequencing the human genome. And the film that you're seeing in the space in the exhibition, I was allowed to use one of my colleagues' Astrofire microscopes in order to make that film. His name is Steve Black, and he studies gastralization and early development, and they're looking at spider eggs in order to understand human development and see the pacing of chromosome splitting and what goes wrong and what goes right, and when it does. He allowed me to make a 200-hour film with him and his assistant. And so what you're seeing is 10 films spliced together in the exhibition, and what I was trying to say with that, if I can cut to the chase with the exhibition, is that some of us live, some of us die, and it's only different pacings. And so what I was watching, as I was watching the development of these spider eggs, is that only a percentage of them made it through the whole series into becoming. And I wanted to have a film that actually documented that and also gives us the reality check of this is a factor in nature. It's not all just coming into being. And it's also quite stunning to watch that development as well. So making an installation, I'm interested in giving a kind of physical, architectural, phenomenological experience. So creating the paper walls like the skin on a cell, or were given to me, and I then edited and worked with them for actually a couple of years by the biophysicist Andrew Pelling, who was working at the time at UCLA's lab, and it's nanotechnology, and this is an AMF microscope that listens to the sounds of cells. And what you'll hear in the film is that cells that are sick sound quite differently than cells that are healthy. The cells that are sick sound like they're stretching, and I think our molecular biologists will be able to speak to this even more than I can, but the use of nanotechnology is amazing because it's a non-invasive way to detect sick or cancerous cells. So that's just a little bit about where my work's coming from and why I think it's, I guess, important to me to know what these people are doing. I use it maybe metaphorically, personally, but hope that it reaches people more globally than just my own story.
Thank you. That really gives us an introduction to Jane's work. And so I think I want to start the conversation with our biogeneticists. And so, Kevin, what's your first sensibility looking at this work, and can you comment on the biogenetic era in relation to what you see?
Yeah. Well, I mean, looking at the work, I'm just struck by, I think it's cool that someone, is taking what we do on a day-to-day basis and putting it into something that's so beautiful and aesthetically pleasing. I mean, we spend tons of time figuring out how to portray the data that we collect, and two fellow scientists. And often when we do that, it's very, concise and accurate, but, we don't take much time to put it into something that we think is beautiful. Often we have these images, like she says, via microscopy and other techniques, and they sit in our notebooks and we'll show our fellow scientists, hey, look at this really cool thing I've got. But they're not really conveyed around to others, and so I think that's what I was struck. Second question, I think, what does it mean to be in the genomic era for everyone? As a scientist, I certainly don't know, and I think probably most scientists don't know. The one thing that's clear is that the technology has, we live in this technological era of genomics, where it actually wouldn't be unreasonable to sequence everyone's genome sitting in this room right now. And, so probably most people don't recognize that. So that can be done. But of course, that just yields us ACGT sequences. I think why I say we don't know exactly is it's unclear how long it's going to take us to translate that into the real basis of a lot of diseases. Simple diseases it already works for. And I think the sense that everyone has, or most scientists, is that eventually that data, all that data mining, will yield us the answer to most diseases, right? It's clearly there in the DNA, in the genetics. It's just, I think, what we really don't know is whether we're going to get there in 5 years or 50 years, depending on sort of the disorders. That's my answer.
So in other words, we aren't there. We are already there. And yet, at the same time, it's very hard for us to grasp it or come to some kind of understanding about it. And as a mathematician, does it ever affect you, this new era that we live in?
My DNA affects me all the time, but that's what I'm told. And directly with mathematics, I can't say on a day-to-day basis it affects my thinking about mathematics, but I mean, as a scholar, we pay attention to a lot of different regions, and for instance, I have tried for years to keep up with Nature Journal and the Science Journal, and it's tough reading. It's pretty thick stuff, so anytime somebody like Jerry can make things a little more accessible or a little more interesting to the people who don't want to slog through the journal Nature, I mean, that's such a great thing. As for mathematicians are making quite a contribution to DNA research, and there are some places. You mentioned that you have some funding, and the Burroughs Wellcome Fund funds the program in mathematics for molecular biology, and a lot of programs in that. I've participated in that where we actually got to go in a lab and splice the plasmid and do other things. I don't know exactly what we were doing, but it was really interesting. But in the short and long of it, mathematically, I am a knot theorist, which is literally, we try to classify knots, which is just fun. So it's pure math fun. But it turns out that DNA ties itself in knots. And I've got, there's a picture of a piece of DNA tied. I think somebody convinced the DNA to tie itself in this knot. This is, honestly, I mean, it's just a beautiful little trefoil knot. But I don't think that's the state that DNA hangs out in all the time. But mathematicians are doing a lot of research, and one of the places where mathematicians are becoming involved is in the action of enzymes like topoisomerases. Topoisomerase, from my understanding, kind of it goes into the cell. Well, I guess I ought to explain why we worry about knots, DNA knotting anyway, but it's my understanding that if, I read this in Colin Adams' knot theory book, that if a cell were the size of a basketball, then the DNA contained inside it would be 20 kilometers long.
Wow.
And that's a lot of stuff to stuff inside something the size of a basketball. So if you can imagine a garden hose 20 kilometers long and you're trying to stuff it into a basketball, it's got these steric considerations, it wants to stay in a certain shape. You certainly can't just crush it down in there, it's not going to be the same shape. So it turns out that DNA puts itself into pretty interesting configurations to pack itself into the cell. And then enzymes like topoisomerases have to break that DNA and unzip it in order to replicate the DNA. And interestingly enough, it seems like some of the topoisomerases remember to reknot it the way it used to be. So there are all kinds of really interesting things, and I only know about these things secondhand, but they're very exciting. Maybe I should get into this a little more thoroughly. But anyway, I could go on, but we should probably let somebody else talk.
Well, since you are the woman scientist here, I do want to know, too, while you're talking, I mean, how did you get into the sciences, and math being at the cusp of going your way, especially the philosophy of mathematics is really a humanities to me. But, you know, I mean, can you remark on that as a woman wanting to go into the sciences?
Well, I graduated from a little bitty high school. There were 8 people in my graduating class in a little country school. And I was good at math, and everybody thought it's a girl and she's good at math and she could get lots of scholarships, so she should be an engineer. So I went to the engineering college to be an engineer and found that that was not my thing. And I actually majored in life sciences. I didn't finish that degree because I was terrified of finishing a degree and winding up in a lab all the time. So I was scared that I wouldn't get into medical school or anything like that, and if I had to stop with a bachelor's degree, I'd be stuck looking through a microscope, which wasn't as much fun in the 80s as it seems to be now. So I started on a backup degree. My math degree was just in case, just to save me from the lab. And I accidentally, literally accidentally, wound up with a co-op job at NASA. And they were interested in the math, not the life sciences, and I was out of money. And when I got close to a degree, I grabbed that math and went for the job at NASA. And so it was all kind of an accidental, but then the pure math is just, I am very much a forest for the trees kind of person. I'm the person who examines the tree and forgets that there's a forest. And so, you know, examining these little problems like knot classification and theory and making sure things fit together right was definitely, as you say, it's kind of a humanities that's solving, it involves quite a creative process, and that appealed to me.
That's great. Thank you, Amy. And last but not least, I want Darrin to talk because, you know, we are a NASA campus, and nowadays you cannot put up a show and not recognize where your show is, who your community is, the people that live here, and this iconic I think only Darrin can talk about.
Thanks, Jane. I'm an engineer, so I spent a lot of time thinking about how I can apply scientific principles. Not necessarily a scientist who's there trying to understand what those principles are, but then once we have a body of knowledge, how can I make something useful for society? I was thinking about how, what does this artwork mean? And what struck me most is about how art and science really, I think, has a lot more opportunity to be mutually beneficial. If you think about how can science benefit art, well, for one thing, it can help artists better understand themselves and their community. Number two, just as we see with this work, it's providing a new medium to express, for art to express itself. How can art benefit science? Well, I think there's a number of really great ways. And one, first of all, just to communicate its results. Trying to communicate science to somebody who's not a scientist is often, as you know, is difficult because they get into all the details of what the phenomena that they're studying. So what's a way that we can put it at a level so that people can understand it? Visualizing the implications. So we just talked about the idea that we can sequence the genome of everyone in this room. Well, what are the implications of that? And I think there's a lot of benefit or a lot of opportunity that art can help visualize what are those implications. Inspire interest in science. Well, that's sort of a self-interest, to get more students to be interested in science. It's not an easy subject, so we need a way to be able to inspire people into science fields. Dispel superstition. If people learn more about science, they learn more about how the world works, they're less apt to believe in superstitious phenomena. And the last thing I was thinking is art can benefit science by exploring ideas for possible applications for new scientific results. Those are some of the things, because normally when you traditionally think about art, some of the things that may come to mind as aesthetics, beauty, pleasure, communicating values, rituals or ceremonies, honoring people, places, or things, enticing emotions such as like color field painting you might have seen, good craftsmanship, or even reflecting personal and societal issues. Those are some of the things that you might think of traditionally as art, but I think there's a lot of opportunity for art to benefit science. I was thinking about specifically to NASA because you mentioned that's where I work. We probably one of the most visible forms of art at NASA are those mission patches. Probably everyone has seen a mission patch. And what is it? It's a piece of artwork that symbolizes the meaning of the mission, particularly for here at the Johnson Space Center. We have our manned missions. Usually the mission patches will have who are the astronauts on the mission, but then it'll also have some symbols of what are the significant accomplishments they're trying to achieve, or what's the significance of the mission. For instance, the last shuttle mission, the STS-135, if you look at the mission patch, it had the Greek letter omega because it was the last mission. So art was playing a role there in the mission patches and documenting and honoring the events for posterity. It was inspiring exploration, and one of our roles at NASA is inspire the study of the STEM subjects: science, technology, engineering, and mathematics. But I think there's a lot of opportunity for art to also participate in communicating vision, exploring possibilities. What are the different applications for technologies that either NASA or any of the other technologies technological companies, and also demonstrate processes that take a long time to evolve or can't be observed through the naked eye, like the processes that she's demonstrating here. So
Great.
that's my initial thoughts.
Thank you. Thank you.
I'm going to ask Gary one last question before I open up your questions. And because I think that we are looking through a molecular gaze now, a reading of the body in a way. It becomes a sort of portraiture for us, a new self-portrait of the cell. It is an identification of the cell. And, you know, the most obvious example of its past is the effects of social Darwinism. I mean, we can really go there with thinking about genetics and expressions of the inherited characteristics and corporeal anomalies have long been prominent subjects in art and literature, and a new conception of race and identity that emerges with her work. I just want her to just comment on that before I open up to your questions for anyone on the panel. Here,
I'm getting used to Jane and what art can do, because science and art are very much linked because we're in physical visual worlds. And I have a lot of scientists who take my classes, and a lot of their work has actually come into my classrooms. Many engineers love to build sculpture. I teach sculpture, I teach bookmaking, I teach contemporary criticism. So we're often thinking a lot about what they're going to go out and do and how they're going to visually and physically make those things. So I really appreciate that connection. So Jane asked me the hard question.
I did.
And I'm hoping all of you could also talk about this too. And I have a couple of images of, so I ended with talking a little bit about, you know, why I made the film and why I made the installation. I'm very much interested in inspiring people to think both formally and poetically about the work. But if you go into the exhibition and at NASA's main campus, I put the images of the egg from its beginnings to right before the spider's legs bud, and I wrote the whole description of every one of the phases, and I worked with the developmental biologist to write that text. And that image is also in the hallway of the biology building at Reed College. So this is an art piece. I see it as an art piece, but it's also a scientific evidence piece. It's, for me, it's a poetic evidence of the mystery of life beginning. But for the biology kids, when they're first learning about this, they can go and count every 20 frames. This is what's happening. So it's kind of a fun play between, I'm really interested in finding out what's going on in the labs. And thinking about, what Jane was asking me about the concept of the collaboration and social Darwinism.
Yes.
I had the opportunity to work with the Department of Medical Genetics at the University of Washington for a couple of years. One of the geneticists took me in his lab and showed me the DNA samples. He studies soft bone tissue and Huntington's. Showed me his mini fridge with the DNA. And then I worked with Arno Tofsky, who started medical genetics. And Arno told me that we are all a tractor-trailer load of qualities and anomalies. And it used to be that we thought, oh, all Semitic people, they have all these markers, or African-American people have all these markers. Well, guess what? We all have all these markers. So I made a piece that was, it's called Case Study, and it very much looks like the Torah. And there's scrolls, and they are hand-embroidered with the markers that I found in my family and my extended family's line. So putting it out there. So, and I didn't have to have the genetic test. This is National Institute of Health's graphics. You can log on, as you know. And I'd love to hear what you think about those graphics, because they're really quite stunning. And you know, any person can go on and say, okay, what I have breast cancer. We think we have the breast cancer gene. What gene would that be on? And you can yourself regulate your own medicine by saying, okay, I'd like number 17 tested. What do you think of that whole phenomena? And you know, because yeah, I'm just really interested in what you think of that kind of agency that we have.
Well, I guess, for the first thing, as a scientist, like I said, it's basically here or it will be here in a few years, right?
Yeah.
Where, like I said, so as we were talking earlier, probably within 2 to 5 years, the $1,000 genome will be here, which means for $1,000, you guys can go out and have your genome sequenced. That will give you, a lot of information, but then you could go back and check and say, okay, do you have this BRCA gene or one of the common sort of breast cancer genes that are well understood, and we know that certain mutations would give you high predisposition to have breast cancer, and this happens for many other cancers as well. And then, so I'd say, outside of, as not playing not the scientist here, but more as a humanist, that's very much the sociological questions you guys can ask yourself. Would you want to know that? And I think that's really is up to, and there will be probably more debates as we learn more of what, how, what we want to do with all this information.
Right.
So, but you'll be able to ask yourself for these things if you have, propensities for certain cancers based on this sort of stuff, based on these tests.
So, it's this question of bioethics that we began to work with a person at the University of Washington, my Genevieve, that is working on writing the ethical discourse for the state of Montana. So how they're going to deal with the testing. And I actually became very interested in DNA testing via a student who came into my classroom one day when they just started doing DNA testing in the labs at Reed. And he brought in his DNA fingerprint, and I was fascinated by that. And so at the time, the Patriot Act had been passed, and I am married to an Arab American, and I decided that this was a really loaded thing, that everybody's DNA could be tested and kept on file and then proved what kind of criminality that you have potential of. So I've been very aware of the slippery slope and the tricky situation that we're in. I made a piece that is very much about that, and was also a piece that I think is poetically very beautiful. It's a rug loom, and it's the DNA fingerprints of my son, his uncles, and my husband's entire male family line. And what I did was I shot the threads through the markers. You can see that my little boy and his grandfather have the same markers in this piece. It's many layers. If you look at the side, it's layers and layers. And the idea that we were all connected was really this beautiful thing to me. And that could also, be potentially used against people also seemed really, vulnerable to us. So I made this piece because I was asked to do an exhibition in the Portland building, downtown Portland, and that's right across from the FBI headquarters. So I thought it would be really interesting to write a little thing about the Patriot Act and what it meant to people. And here's my husband and son. I didn't say what our names were, to give the full details, because there they were, right? They could, incriminate us. So, we were talking about what art can do, and I was really thinking, I think a lot about my work as a mirror up to our society and saying, hey, look at this, guys. We can do this. We have a lot of power in our hands right now, as citizens, to say, no, we don't want this, or yes, we do, And that said, I'm not on the, I am actually a proponent of genetic testing because of my own medical experiences, right? So, but I do see the ethical situation. So that's another view of the piece. But I just thought I would, since you asked where I think it's going. Genetics, it's a public artwork. This is a close-up that's hand embroidered. All of the major discoveries, and it's chromosome number 17, which the breast cancer gene and the ovarian cancer gene was discovered at the University of Washington, and that's on 17. But what I just wanted to conclude with by saying, I'm going to splash through a couple of pieces, is this is chromosome painting when we first made it at the Kirkland Arts Center. And what this piece is the chromosome being injected with phosphorescence, and it's done to do a comparative analysis to see if you have anomalies and what they were. It's old technology, it's 10 years old. So, what you're looking at over there in the galleries are these chunks of color And they literally were the chromosomes that had colored stripes on them from the phosphorescence, turned into a computer program so that it could be charted out. Am I explaining that? It's old technology. They don't use it anymore, am I understanding? But I found it to be really, really beautiful, and Arno gave me this beautiful synteny map, they're called. And what I ended up doing is not only making the installation, but also I started a textile company called GoForward.com, and we are raising money for people to have their genetic tests done. They don't need to contact their insurance agency. We will just give them the money, and they can have their one gene tested if they believe that they have inherited the ovarian cancer gene. It's at the University of Washington. And some of those pieces are upstairs, but there's a website, and we've sold a number of them and raised some money for people. I work directly with a genetic counselor to do that. So it's kind of the idea, I think artists often stay in their studio and make nice things, or interesting things, or comments like I made with the DNA piece, but I really felt like I wanted to take this out because I wanted to tell people, you'd probably rather know and do some preventative stuff than to have surgery. So I was very interested in that aspect of it. So these are shots of the installation, and then the scarves that are on the light boxes, and those are touring a couple of other locations this year as well. So I am a positivist about these things, rather than I think a lot of people have a kind of fearfulness or a thought that science is going to be the destruction of us all. So, anybody else want to comment on these things?
Anyone has a comment at this point? Before I open it up to the audience, I love the remark that we were talking just the other day amongst ourselves to think about genetic testing making surgery obsolete, making the sort of invasive kinds of treatments that we have today seem like butchery. If we can eliminate them, but through prognosis, through diagnosis, instead of going through bad, hard treatments, that's what we are looking at for the future, I think. So let me open it up for questions. Anyone? Meryl.
All right.
And who are you addressing?
I guess Dr. Ondrizek?
Just call me Jerry.
Okay, thanks.
These guys have PhDs.
Okay. I like what you said about the fact that we operate in visual and physical worlds. And as a result of that, I was curious about how you used dimensionality in your works. Were there some works that you prefer to make one-dimensional over others? And if so, why?
Well, I guess that because I got to use those really cool tools that scientists use, and I realized that chromosomes aren't flat. They weren't drawings on paper, and that the knots are really amazing. Your knot theory things, it's a spatial situation. They're all tangled up inside there.
Is it four-dimensional? Is it four-dimensional or three-dimensional?
Oh, well, we knot theorists study our knots in 4 dimensions just so we can make them do things they shouldn't do. But, no, but I mean in the real world, in the DNA in the cell, they have to stay in 3 dimensions.
They
Well, I guess I was just wondering because if we want our audience to interact with science, it seems like people interact better in multiple dimensions. So I was wondering if there was an audience component.
Right. Well, I guess that as a sculptor, I've always thought about making the physical world, and you saw some of the greenhouse and the early furniture that I built. Definitely interested in architectonics and phenomenological experience. So I do believe that we learn through kinesis, we learn through touching, we learn through feeling, and that every material has a semiotic. It's something, it's telling us. So I'm very careful about what it is that I'm physically using to make it and how big and the scale of it. So the chromosome paintings, for example, that piece was the commission for the Kirkland Art Center. It was a grant from the Ford Foundation, and I designed the panels, the size of the panels, to mimic the timbers in the ceiling of the 100-year-old building. Wow. So So that's why you're seeing them that size. And those timbers, of course, are, about the width of our bodies, the space between. So I was very much thinking about the chromosome as a body itself, right? So you'd stand in front of it and kind of have that relationship. And the same with the cell projection. You'll see that projection, you stand in front of it and you're in the cell. Like you get up close and personal. Your head's like the size of the chromosome splitting. So you can really see it, and it's not like just a fantasy that mathematicians are telling us that they're nodding up. You could actually look at them and see that's true. So it's kind of what the NASA people are doing by taking the telescopes all the way out, right, and showing us these things. And we, in our fantasy, we believe, oh, those planets are really close, but they're really far away. So, yeah.
Yes, Martha.
So I see these images and this is a question to all of you as experts. I personally see in communicating this new, I can use the term new knowledge, new to me, for example, in mass production, okay? And so I'm coming to ask, for example, I see those pieces as like shawls or wall hangings or even, I don't know if this is going to be disrespectful to you all, this is what I'm getting at, a table liner, something that...
You can use it that way.
So how many of you all see that as really being low rent? I mean, you're wanting to communicate, the mass communication, and so do you see that though as like lowbrow, pretty low rent? That's not how you want to communicate this versus an exhibit.
Martha, are you suggesting that the decorative factor reduces the meaning? Is that what you're trying to say?
Yes. I'm wondering if that, because that's the way I would communicate to the world. This is something that's a value.
But then on the other hand, does it make it more accessible? Is it more...
If the message is the thing, she seemed to be, then is Jenny?
Yep.
It seemed to be in her heart, the desire to message this, to communicate this. So, in my background, I'd put it on a coffee mug, or make it accessible.
Accessibility?
Yeah.
Or,
Well, I mean, I would be interested to hear what other people think, but for me, I was very purposeful about making it accessible, because I think that very few of us take genetics or knot theory or become NASA engineers. We don't really know what all of these people are doing, right? And nature genetics is very hard to slog through. So that synteny map test I thought was so magical. And it's actually what happens when they inject I didn't make up the colors, I swear. That's the colors that happened. And those colors indicated the levels of proteins and markers and where the markers lie. So I translated it. I hand-dyed the silk, got the colors just the way I wanted them to match, right? And then I use a company in North Carolina that print them for me. And I am giving 50% to medical genetics. So I'm not making hardly anything on these because they're hand-sewn, they're silk, they're 100% silk. And it's not about making money for me. It's not about me being famous. Anyway, it's about getting that message out. And people have suggested to me to put it on a coffee mug, but I have this real love for cloth. And I think, we wear clothes every day, and it's our second skin. And, Jen and I were talking recently about this factor. When we have loved ones who are sick, we want to wrap them, we want to hold them. And these are pieces of cloth that are badges of honor. And they're something that as you're suffering that cancer to wear, not unlike the pink ribbon. And it's been interesting to me making that work because it brings up a lot of emotions for people. I had a whole group of breast cancer survivors in my studio recently, and my mom died of Hodgkin's disease, and I've kind of lived my life like wanting to make a difference and think about that, and I inherited that marker, which is also something I think a lot about. And I think it's almost a bonding factor. At the exhibition, I met the woman who's designing my website now because her mom died of it. It becomes, in some ways, there is a breast cancer club. It's true. And I think that we have to face the fact that we do have these things that we can actually support each other in and maybe even help each other somehow. So that's a mission, and that's different. And I hope it's not lowbrow.
I like your idea of wearing it as a scarf, as a badge, because, I mean, in education we think a lot about different learning styles.
Yeah.
And the tactile and the visual and all these different learning styles, and the idea of being able to feel the silk is just, that's just another dimension, I would think, to actually be able to feel it and see it and touch it.
I think Dr. Gladden has it.
Oh, okay.
Well, my question kind of follows the previous one, but it really is for the scientists on the panel. Accessibility is about issue of audience, and I've been thinking a lot about who the audience is for this exhibit. I know a lot of us who have heard the presentations about your work from Jane and from Jeff are taken with this notion of the sound of cells dividing. And so a lot of us who have heard that are more from the arts side of the picture, from the science side. This is an amazing discovery for us that's very exciting for us. We can't wait to hear about this. And so for that audience, it seems that what's happening here is that art can teach us something about science. For the scientists on the panel, for that audience, my question for you is, what does this art teach you about science? What do you learn about the work that you do and the things that you study when you look at this through the perspective that Jerry's work brings to you?
That's a great question. Great.
Well, I think most of us who study science, and we pure mathematicians definitely feel like it's art. And we, you see this beauty all the time when you're working on things, but then to see it up, and accessible to other people, it is a reminder for the scientists that maybe we need to present more, maybe we need to make things more accessible instead of hiding it in our journals and writing long papers that nobody's ever going to read. Anyway.
I was just going to add to that. Earlier I had made some comments about what are the benefits, what art can have benefits for science. And as Amy was saying, you do your science in a sort of a closed environment, and so communicating its results, you're usually not the best person to communicate that out to the people at large. So I think artists.
Why would you say that, Darrin?
Well, I'd say
You're not the best
because
person to
you're the expert at this particular topic, and you make a lot of assumptions about your knowledge, and others seeing that or the artists particularly will be sensitive to what are the things that a person looking at this that isn't an expert at it needs to be able to see to draw conclusions about it. So visualizing the implications of, that's another thing, if you're dealing in pure science, what are the implications of all the work that you're doing? You know that science tells you how nature is. It doesn't say how nature should be or how you should use the science. So what are the implications? And I like the idea of inspiring interest in science, because if you're in the lab, you're working with test tubes or any of the other implements of science, it's not necessarily the best way to try to inspire students to study science, because you want to understand, for them to understand why is it exciting? What are the things that makes you excited about coming to work and doing your work? So my thoughts.
Kevin, do you have any remarks?
Sure. Should I? Well, I guess this has certainly made me think, and I'd say almost by definition, at least the scientists who do what I do, we are kind of our job is to be reductionists, to take very complex living systems and turn them into very, very simple, simple things. And so when we convey our results or data, there's sort of, it's an unwritten rule, it's conveyed in sort of as simple and concise a format as possible, so it doesn't have to look pretty, and it's only for sort of our immediate audience, right? So like, simple example is, if black and white can do, you do not publish in color. And the other funny thing is, well, I think people are recognizing things. I don't know how, if you guys know how scientists convey information today. We do it via print journals. It's completely archaic, right? It's absolutely crazy in this day and age.
You have the wildest technology to make the images.
Yes, and it is. We have all this crazy equipment and all this, like you're saying, all these amazing toys, and we get all this stuff. And I think people are talking about this, that this will change, but we still convey it in print journals, in generally simple sort of black and white images that are meant for me and the guy across the hall to communicate with each other. And it's not meant to go to the lay public. So I can tell you what I've been thinking about is, it does make me question if, like, when I give a talk or the next one, with some of this data, could I somehow convey the same information and yet make it a little bit more accessible to some members of the lay audience that are out there? And it's got to be possible. It's making me rethink, how to present that data. Could we do stuff in a little cooler, more artistic fashion that would also still convey the science, but also make it maybe a little bit more accessible to, who might also be out there in the audience.
And Walton, and then maybe one more question, and then.
I wondered if the dual panel installation here at the back door of NASA brought about any ideas of parallel universe.
Physics people?
I'll be brave and take this. But I'm going to take it in a little bit different direction, though, because I was thinking about this. It's like, not parallel universes, but a parallel idea that, when you look at the cell, you're taking a microscope and you're looking at something that you can't see with the naked eye. So you're using a microscope, you're using a tool to be able to see it. On the flip side, on the grander scale, looking at stepping back and being able to see, let's say, our home, planet Earth. Because in a similar way, you're seeing humanity. This is where we live. In 1968, for instance, was when Apollo 8 first took a picture of the entire Earth. And people were able to see, this is our home, this is planet Earth, and it brings a different perspective on humanity. Just like looking at a cell brings a different perspective on humanity. And then, in that same mission, Apollo 8, they were able to take an Earthrise photo. It's an iconic photo, so most people have probably seen it. The Earth is rising above the moon's surface. Fast forward to 1990, on Voyager 1, Voyager 1 took a picture of the Earth, the furthest that we've ever seen ourselves. And that was about 3.5 billion miles from Earth. So you think about images of yourself, images of what makes you, and it could be cells, or you could be looking at it on a more grander scale and looking at where do we fit in the universe. So kind of drawing a parallel between looking.
That's great.
Yeah, that's a beautiful comment. Jane and I were talking the other day about this too, and I was saying that our generation has lived through the space race. We were on a race to get to the moon in 1968, and we were on a race to get to the genome, and we've gotten to see those two spaces. And I think that similarly, similarly the space race and the race to do the whole human genome are very similar, and they have a similar kind of effect on an aha moment societally, that we saw something fully for the first time.
Right. So I think our last question should be from Dr. Subramanian, and then we welcome you to come and meet our fabulous artists and our fabulous panel
I guess I don't necessarily see it as being critical to my work. I guess we could continue to have our little narrow alleys that we teach in, but certainly it can't help but expand understanding, and I would like to see more of it. I guess I should drive for that.
Well, from the NASA perspective, one of our missions is public outreach, education of the public for science. Tell the story of what science is all about, and study science, technology, engineering, and mathematics is going to make us as a nation more competitive. So that's, I think it's absolutely critical, particularly from my perspective.
Well, I mean, so for the work I do, or anyone that does biomedical research now, at least in a scientific sense, it's completely interdisciplinary. There's no boundaries. I mean, like genetics in point, I mean, started off as, say, basically molecular biology, but that takes the work of tons of mathematicians and computer science. Biomedical research is completely an interdisciplinary game right now. And so, and I think in terms of maybe for this panel and, you know, artwork like Jerry's, maybe not so much, but I can definitely see it. I mean, the main thing for me, or to do the type of work we do, is incredibly expensive. A small lab goes through millions of dollars easily. So it's making, back to the theme of sort of accessibility and getting stuff across, I think that's being able to convey what you're doing and making it accessible is incredibly important, because you've got to convey what you're doing is important to continue to fund the research you're doing. So yeah, that's kind of my thinking today of, obviously, the more accessible you can make it, the more you're going to be able to continue to fund the research you're doing.
So I just mentioned that I just wrote an NSF grant with my colleague at Reed, who's a biologist, and it's a grant to do what you're talking about, and I'm just there really to help coordinate to get visuals attached. So she wants to do cross-disciplinary, cross-scientific disciplinary. You know, it's an engineer who made the AMF microscope to feel the cell, and it was a physicist who figured out how to turn the vibrations into sound, and it was a biologist who used it to determine if it was cancerous. So it is so interestingly cross-disciplinary, and in terms of the chromosome painting, that's a mathematical program. You know, it's a computer program. So statistics comes in, and you know, we're really, with this grant, trying to reach out to all of the different majors at the college I teach at, but also we'll reach out to the medical school and the other universities, so we have cross-disciplinary.
Well, thank you so much for coming. Come and hang out with us in the gallery. Be sure to experience this work. That's the only real way that you'll know